So if you're year 13 A-level biology student and you are trying to revise glycolysis, then this video is for you. So glycolysis occurs in the cytoplasm and it is the first stage of both aerobic and anaerobic respiration. So the first thing we need to talk about is what actually happens to that glucose molecule. So we know glucose is a 6-carbon molecule, so I've drawn it here as 6 Cs. Now that glucose molecule is going to be phosphorylated. So when something is phosphorylated, what we're saying we're doing is we're adding a phosphate. Now, if you're studying AQA, you need to know it as a two-step process. So glucose is phosphorylated to glucose phosphate, and then that glucose phosphate is phosphorylated again to hexose biphosphate. If you're studying for OCRA, then you just need to know that the glucose is phosphorylated straight to the hexose biphosphate. Now, this 6-carbon molecule, which now has 2 phosphates attached to it, is a very unstable molecule. Now, because it's an unstable molecule, it will automatically lyse itself into 2 triose phosphates. Now, that's a triose, tri meaning 3 O's, 3-carbon molecule attached to a phosphate. So that triose phosphate then, the next thing that's going to happen to get us down to our molecule that we need at the end of glycolysis is we need to have oxidation occur. Now for oxidation to occur, what must happen is that triose phosphate is going to lose 2 hydrogens. Now those hydrogens are going to be picked up by these coenzymes. So what you have is NAD plus is going to come in and it is going to pick up 2 hydrogens from the triose phosphate. Therefore, it's going to oxidize the triose phosphate and the NAD, the coenzyme, is going to become reduced. It is reduced because it has picked up hydrogens and electrons. Now, I like to think of coenzymes a bit like taxis. Taxis that are going to pick up a substance and they are going to deliver them and drop them off somewhere else. So our NAD coenzyme is going to pick up our 2 hydrogens and it is going to deliver them to where they need to go. And we'll talk about that when we do oxidative phosphorylation, but that's a whole other video that I will put on here. So the key thing to remember is that the triose phosphate has been oxidized whilst the coenzyme NAD plus has become reduced. Now, not only does the triose phosphate become oxidized, what also happens is we're going to remove some phosphates from it. So to do that, we're going to add phosphates into ADP, adenosine diphosphate, with an inorganic phosphate attached to it is going to become adenosine triphosphate. So here you can see I've got what's called substrate level phosphorylation. I have phosphorylated my ADP to ATP by using a substrate. Whenever a phosphate group is transferred directly to another molecule, that is known as substrate level phosphorylation. So I always say to my students, it's really important to go back and look at what was the input into glycolysis and what's the output. So what did we put in? We put glucose in, and what did we get out? We got out 2 pyruvates. Now, what did we put in? Well, we put in 2 ATPs in that first phosphorylation process, but at the end, we got out of the substrate level phosphorylation 4 ATP molecules. Well, that gives us a net. We had to put 2 in, we got 4 out, so overall, we have 2 ATP molecules being produced. We put in 2 NAD plus in the start, and we got out 2 reduced NADs. Now, we can call that NADH plus H plus, or it can just be called NADH. So it really depends on your exam board what it is that you should be using when you look at this. But I always tell my students, if you're not sure, just write reduced NAD, because that refers to it having picked up the hydrogens and the electrons. So you don't have to worry about how many there are. If you are writing it, just write reduced NAD. So the key thing I always say to my students is to look at the input and the output when it comes to each of the different stages of respiration. Now, glycolysis is the first stage, which is anaerobic, and our input is glucose to give us the output of 2 pyruvates. Now, we need to put in 2 ATP molecules. The output is that we get 4 ATP molecules. Now, it's really interesting because we have to put 2 in to get 4 out. So overall, we end up with a net 2 ATP molecules. We put in 2 NAD+, and we get out 2 reduced NADs at the end. Now, if there is no oxygen present, the pyruvate will continue down the anaerobic pathway. Now, if that is in animals, then that will be the lactate pathway. If it is in plants, it will go down the alcoholic pathway and produce ethanol. If oxygen is present, then the pyruvate will continue through the next stages of aerobic respiration. And to do that, it must enter the mitochondrion.